name is Fedor, and this is my project, purification of EGFP in brovykh 2.1. As you see, the picture on the top is the EGFP. It's green fluorescent protein. It comes from jellyfish, and basically it shines green, whatever you see. The picture on the bottom is brovykh 2.1. My job was to purify this construct. All right. So the goal of the lab where I work, which is the Green Lab at Columbia University, is to study fundamental interactions between protein and nucleic acids and to learn more about cancer-related proteins. And actually, brovykh 2.1, the picture on the bottom there, is very important when it comes to cancer research. So my project, my overall project consists of these 4 major steps. The first step is transformation. The second step is grow colonies. The third step, regulation of gene expression, and the fourth is purification. I will explain them as I go along. All right, first is transformation. What is transformation? Transformation is introduction of exogenous gene into a bacteria. In my case, the bacteria were BL21s. There are a lot of cells that are used in research. Some are DH5 alpha, but BL21s are distinct because they tend to grow slower. My construct looked something like this on the picture on the bottom, and my job was to chip kind of just purify this, so eliminate the GST tag and the his tag.
So what are we seeing there? What is it?
Oh, this is a piece of a plasmid that we see.
What is plasmid?
Oh, plasmid is basically DNA in bacteria, which cells cannot take. And
what are those boxes?
The boxes are the tags.
What are the tags?
Tags are pieces of DNA.
Great. So he's talking about pieces of DNA, and those pieces of DNA are called those 4 different things. RAD51.
Actually, this construct is a small particle of plasmid right here. So technically, the plasmid goes all over that. All right. So the first step, growing colony. First step is to make LV agar for plates, which is the 35 grams per liter. What is LV agar, if you ask me? Well, I'll tell you. It's a liquid which, when you heat it, you can pour plates, like the picture, or the second picture, right? And then after an hour, it solidifies and it becomes this jelly-like substance, which then you can spread the cells on, and the cells grow, like in the third picture. So you have to put antibiotics into LV agar because this plasmid right here, which contains my gene, has also a carb-resistant tag. Therefore, the bacteria, the cells that do not accept the plasmid, they die off immediately. Therefore, it's like sort of a natural selection where I eliminate all the cells that I do not want. All right, after like half of hours, LV agar solidifies, and then you can wipe one colony, or you can basically spread the carb first, and then you take the cells, which in my case they were pre-manufactured, and you spread them on the cells, and you put them into the incubator at 37 degrees overnight. The next day you return, you get these dots. You can use these plates to start a starter culture. How do you do that is you take a pipette tip and you wipe the pipette tip with one colony right here, and you put it into LV. LV is basically liquid form of LV. Now, for my project, I had to make, all right, listen to it here because there's a lot of numbers. 4 liter, 4 liter, 8 flasks. All right, you get that? So 4 flasks. I know 8 flasks, and each flask is 4 liters involved. Into each flask, I had to pour 1 liter of LV, which is liquid. right? And then into each of the flasks, I have to put 1 mil of carb, again, natural selection. And then into each of the flasks, I have to put 12.5 mils of starter culture, what I did previously. Then you put all the flasks in the shaker. Shaker is basically an incubator, but the base of it shakes like that, and it helps cells grow. After a couple of hours, we have to wait till the cell density reaches 0.6 to 0.8. Now, what is cell density?
Sure.
Over time, cells grow, right? So per same volume of LV or whatever media you have, more cells accumulate. And you check the density of your cells using a nanodrop. Nanodrop is the machine you see. Now, you put your sample in the prism over there, on the rectangular prism, and the light shines right through the rectangular prism. And the way that the light is reflected or received on the other side, that's the way cell density is determined. After that, you induce it with 400 millimolar IPTG. Now, IPTG is a toxin which basically results in a gene to produce the product, because every gene has a respective product. Like RAD51 gene has RAD51 protein, which encodes it. So when you put when you induce your media with IPTG, you get more protein. Now, an important skill which I learned is SDS page. It's how to test whether you have the protein in your sample. And you're supposed to there's a whole procedure. You're supposed to learn how to make your own gel. It's like gel electrophoresis, sort of, but not quite. So first you take, before you induce, you take a pre-induction sample. And after you induce, after a night, you take post-induction sample. And then you load them onto the gel. How do you do that? Well, you take 1 mil of each sample, right? You spin them. And after that, you get media and the pellet. You suck up the media, and then you stain the pellet with 4-XSDS dye and water, which after you load onto the gel. You attach the cathode and the anode, thus making the electricity flow through the gel. And after an hour, you get something like this, the gel right here. And through these lines, you can determine whether there's protein or not. Now, the last step to my project was purification. Once I did the SDS page and I figured out that there's protein, now I can purify them. I take the 8 liters that I grew, and I spin them down. And this looks like a falcon tube, but really I had a really big container and a huge pellet, which I resuspended in lysis buffer. Lysis buffer, what it does is when you mix it with the cells, it sort of pulls apart the cell membrane, right? And all the protein, those organelles, are now free floating in the supernatant. Now, for me, I had to go home, so I froze it, and I put it in the -80 fridge, but you can still consume it. The next day, I thawed the pellet on a sonicator. Sonicator further sort of does the work of the lysis buffer. It further diminishes all the bonds that exist between the cell membranes. After that, you centrifuge, right? And then you get the pellet and you get the supernatant. Now, the pellet now contains all the cell debris which you don't want, so you throw it out, so you save the supernatant. And then you take that supernatant and you pass it through the actin machine. Now, at this stage, I actually have this construct in solution, right? So, what does the column do? Oh, the column basically pulls away this small piece. So when the solution goes through the actin, all I have is a few atoms. And then the last step, I add ammonium sulfate, which pulls away the last GST tag. And then I have purified VAT51 and E2. And the thing is, I did it last summer, and I came back this summer, and I've been told that people who have PhDs cannot repeat what I did. So I'm pretty impressed with what I did. Further work. Now they put me back. They put me back in the team to purify EGFP and VAT51, and later we can use that to in cancer research. My references and acknowledgments, and I will take questions now. Yes, sir.
All right, could you please go back?
Get up. Can you please get up?
All right, so the SDS page, where you were saying it's similar to gel electrophoresis.
Yeah.
And add a bunch of, that. Can you please that up for a second?
Questions? Yes, sir. Oh, man. Sorry.
Your goal was to purify the proteins, but how is this going to contribute to like science around the world? Like, what's the goal?
VAT51 actually, how should I say? It recovers the breaks that exist in DNA. So it's not directly connected to cancer research, but through various steps you can apply it to cancer research. I haven't been told the whole picture, really, but that's me.
So your whole methodology is just purification?
Yeah, basically.
You just purify the protein. What do they do with the protein afterwards?
They do whatever they gotta do. They have the whole. They have their own project, and they have. The thing is, so far we haven't purified. So.
But if you find out why they do it,
Yeah.
that's a good question.
Yes.
Can you repeat why you did the research?
Why I did it?
Yeah.
Okay. So basically, before the induction, right, the protein doesn't really grow. However, after the induction, after that I add that ITTG, that toxin, the protein grows extensively. Therefore, after and before we see the difference in our protein, so we can determine whether the protein